Hello, kids, and welcome to DNA Structure and Function.
We are your hosts, Miss Pranky and
Mr. Harmson. You might sit back and wonder how DNA is condensed. Well, you're in the right place because today we are going to learn how DNA is condensed.
Yes, we are. So the very first thing you need to do is actually pause these notes and watch this video. And on your note sheet, please fill out how DNA is condensed. So now you should be coming back from the video and filling out your note sheet. And we are going to talk about the building blocks of DNA.
As you can see, DNA is a type of nucleic acid. Hence, its full name is deoxyribonucleic acid. DNA is a polymer built from monomers called nucleotides.
This is a good reminder from our biochemistry unit. Remember, monomers, the prefix mono means one. So we have one nucleotide, and the polymer means many. So DNA is going to be many nucleotides paired together.
A nucleotide has three parts: a phosphate group, a sugar, and a nitrogenous base.
So here you can see these are going to be our nitrogenous bases. They're going to be listed as either A, T, C, or G, which is adenine, thymine, guanine, and cytosine. A and G represent your purines. You can see that they are double ringed here. And T and C represent your pyrimidines, and they are single ringed. A is always going to match with T, and G is always going to match with C. And we know this because of the wonderful scientist Chargaff. That is his rule.
Okay, so we're going to take a look at a DNA strand. The nucleotide monomers are joined together by covalent bonds. Nitrogenous bases are called bases for short. Obviously, as Ms. Pranky stated, Chargaff's rule, A and T are the base pair, and C and G are also the other base pair.
Something that we want to point out to you guys here, you can see. So this part right here is going to be called our sugar phosphate backbone. One of these sugars, one of these phosphates, and one of these nitrogenous bases make up a nucleotide, and they're all linked together by covalent bonds, which we talked about in our biochemistry unit. Those are a nice strong bond, and they are what make up one strand of DNA. As you can see here, now we doubled it. And a couple things that we want to point out to you. If you look at the picture on the left, you can see this blue ribbon. This is going to be what we call the sugar phosphate backbone. So they're not labeled in here, but there is a sugar and phosphate bonded together by a covalent bond in here. And then you can see on the inside, we call this rungs of a ladder. DNA kind of looks like a ladder. It just spins. And these are going to be our nitrogenous bases. If you look at the middle picture, you can see our adenine and thymine are going to be paired together by a hydrogen bond. So remember, we studied hydrogen bonds in biochemistry as well, and these were for water. They're relatively weak bonds, so this is really important for DNA replication, which we will get to later. And then one more thing we want to point out to you guys is the orientation. We know DNA is anti-parallel. So if you look on the left side, you can see the oxygens in your sugar are facing up. And if you look on the right side, you can see the oxygens are facing down. So you can tell here that these two DNA strands are going in opposite directions, which makes them anti-parallel.
What will the complementary base pairs be?
So on your sheet, you should pause this video right now to try and figure out what the complementary base pairs will be for this strand of DNA. So remember Chargaff's rule and write it on your base or on your note sheet, and then come back and check your answers here. So we know A should be paired with T and C should be paired with
G.
Yes. So we call this our template strand on the left
And our complementary strand on the right.
because they complement each other.
So now we're taking a look at clues to dimensions and structure. As you can see, this black and white photo looks really familiar to the movie we saw for the X-ray crystallography. And Rosalind Franklin and Maurice Wilkins produced that X-ray into crystallized DNA, so we can kind of get an understanding of what that helix looks like. These diffraction patterns in the picture provide clues to the basic shape and dimensions of DNA as being a double helix. A 1953 DNA structure modeled. And I think you guys know that before because the movie outlined that really well. So James Watson and Francis Crick have obviously been hugely recognized as discovering the structure of DNA. Why do you think that finding the structure of DNA was so important?
It's important because it tells us how things are passed on from parent to offspring. So it really helps us determine how heredity happens in organisms. It is how we store all of our information in each of our cells. So, in order to grow and develop, you need to make more cells. And it also tells us how organisms can be so vastly different from each other and how some can actually be genetically identical. So, DNA is like the blueprint to all life on Earth. So, what we are going to do now is we are going to practice. So, you guys need to work through these problems on your note sheet and then submit it when you are done.
Perfect.
Have a great day, you guys.
Wonderful. Hi, though, for real.
Bye. I don't know how to turn it off. Wait. Wait, does it say? Oh, man.